Here's where we left off. We were talking about the binding rules of complementarity, and that explains why the two strands of DNA fit together like a puzzle and are joined by hydrogen bonds. I told you one way I think about them is like a ridiculously easy jigsaw puzzle, and the other way I think about them is like a mold that has got two parts. So follow me on my analogy. Let's imagine that this is something really special, some sort of antiquities kind of thing. And so this is something that your museum has got, and you want to be able to sell copies of them in the gift shop. So you're going to sell copies of this in the gift shops for people to take home. How are you going to get it done? All right. Well, one way to do it would be to give this piece to a set of artisans and ask them to carve duplicates of exactly that piece and give that to a different set of artisans and do the same thing. Right now you're thinking, Tidal, that's a dumb way to do it, right? Of course it is. This is a piece of pottery and a mold. The way I would make more of these guys would be to give this to maybe a single person and ask them to just put clay in there, let it harden, fire it, glaze it. I can use this piece to make hundreds of copies of that. And they would be perfect, not like a little bit off. Same thing over here. If I wanted to make copies of that, I would just give this piece over here to someone and say, put clay on that, let it harden, and I would instantly have copies of that. What that means is I can make perfect copies of this side on the right by just using the side on the left. And that is what the enzymes that do this job inside of your cells, that's what they do. They've got two sides, little piece of pottery, little mold, fit together, complementarity, right? And then there will be an enzyme called helicase that pulls them apart. And then there will be an enzyme that will walk along here and will use this to make a perfect copy of this one without ever looking at it. Why? Because when they got separated, now this one is going to be TCTAC, right? Okay. And then it'll just walk along, and wherever there is a T, it'll put an A. Wherever there's a G, it'll put a C, and it'll just work its way down. It's kind of ingenious, right? So what is the reason for all of this? Let's stop for a second, and let's stop to think about like what is the problem that life with a capital L is trying to solve. Here's the problem. Here are the set of challenges. You're not supposed to use the word problem, right? Okay, problems. Right? Here's the problem. The problem is we need to have instructions for how to build a perfect Tidal, as if Tidal is perfect. Okay, but one of these. All right. We need those instructions. Now, those instructions were given to this organism as one cell. One cell had one copy of the instructions to make a Tidal. Right? So every time the cell divides, mitosis, right? Every time the cell divides, it needs to make a copy. A copy gets made. Ooh, that's a problem. Mistakes in making those copies are called mutations, and mutations, I mean, they might make it so that I can fly, but there's more of a chance that they'll make it so that I'm going to die or get cancer, right? So we can't make any mistakes. Every time the cell divides, and by the time I'm me, it is divided more than a trillion times, because I'm about 10 trillion cells big, and many of my cells got left on surfaces along the way, right? So if I only had cell division a trillion times and I have 1 in a million mistakes, I've got a million mistakes. I got cancer, I'm dead for sure, right? So we need to make lots and lots of copies without any mistakes, without any mutations. That's problem number one. Problem number two is we need to have these instructions in a form that can instruct how to build a protein. You know that proteins are built out of 20 different amino acids, 20 different amino acids. These are the problems that life is trying to solve. And this system, molecular biology, manages to make it ridiculous easy to do things properly, hard to make a mistake. This system makes it so that when an enzyme that was building DNA has made a mistake, it's easy for another enzyme to catch that mistake and fix it before it becomes a permanent mutation. And this system also makes it so that even if a mistake leaks through, and even if there is a mutation in the DNA, it is possible that the protein will be properly made. Okay, that's the problem. Alrighty, so nucleic acids. So far, we've talked a whole lot about DNA, not very much about RNA. Let's talk a little bit about RNA and the comparison of them. Because on your quiz, there will be a lot of that comparison stuff, okay? DNA is made out of nucleotides, RNA is made out of nucleotides. Okay, wait, that's not a way that they're different. The difference is that a nucleotide of DNA uses the sugar deoxyribose, and a nucleotide of RNA uses the sugar ribose. Listen to me. If I ask you if there is ribose in DNA, you will say no, there is not, right? Oh, but the word ribose is in there. And you know what? That's like Justin Bieber's name is in the I Hate Justin Bieber fan club. It ain't the same thing, okay? Deoxyribose is a different sugar. How is it different? Right here, do you see how there's an OH? And here, there's just an H. That's the difference between ribose and deoxyribose. Deoxyribose, deoxy, lost for oxygen, right? So that's one way. Another way, DNA is designed to be permanent. DNA is designed to be permanent. How permanent is it? It's so permanent that we can get DNA from mastodons that died 10,000 years ago. They've been preserved in permafrost, but we can get DNA from them. Enough that we think someday we'll be able to clone, not a mastodon, a woolly mammoth. Yeah, they're different. Yeah, don't ask me, I'll tell you. Okay. So DNA is made to be permanent, RNA is made to be temporary. DNA is so permanent that 10,000 years mastodon, that if someone died in the Mojave Desert 50 years ago, we can still get DNA and identify them. How permanent, how temporary is RNA? If I have some RNA on my finger right now, oh, it's gone. Okay, falls apart super easy. Okay, that's not a problem with RNA. That we've designed RNA to do that. Bases, DNA, Gattaca. RNA, everywhere that DNA would use a T, RNA uses a U. All right. So Gattaca was a good name for a movie. Gauka was a terrible name for a movie. They went with Gattaca. All right. DNA is double-stranded. RNA is single-stranded. Let's see. How else are they different? No, I think that's pretty good. Which sugar they use? Temporary, permanent, double-stranded, single-stranded, Gattaca, Gauka. No, that's about it. Okay. Now, the RNA that we're going to talk about, there are 3 kinds of RNA that we're going to talk about in our story, and we'll get to them. So why a genetic code? This system makes it easy to copy nucleotides, right? Those are the monomers, with great fidelity. That means no mistakes. Each amino acid has a 3-letter codon that's been assigned to it, a genetic code, a genetic code. DNA is all kinds of G's and A's and T's and C's, right? Gattaca, we'll just spell Gattaca again, right? Well, that's swell. We know that it's really easy to make the other side just by copying the rules of complementarity, right? So that's great. But here's the problem. Those That Gattaca stuff, that is the instructions for how to build a protein. There are 20 different amino acids that are used to build proteins. So how does this Gattaca nonsense help us build a protein? Because the arrangement of those G's and A's and T's and C's is code. It's a literal code. When we talk about the human genetic code, we're not speaking figuratively or poetically. We're speaking literally. That's code. It took us a long time to figure it out. We were looking at the DNA with all these G's and A's and T's and C's, and we're like, whoa. And then we look at proteins, and there are 20 different amino acids, and we're like, how does this all go together? Because every little three-letter, I'm going to call them a code word, but technically they are a codon. Every little three-letter codon, like GAT, that is the code word for one of the 20 amino acids. Right? Codons. Right? So, yeah, I told you guys that I discovered a protein, right? And this is my protein, right? Do we look alike? My baby. So here in the single letters that are farther apart, you'll see lots of letters. You don't need to know what they mean. Each one of those is one of the 20 amino acids that builds my proteins. Proteins are polymers that are made out of monomers called amino acids. So these are the amino acids. And I'd like you to notice that right above the amino acids, there are three letters. Like, oh, sorry, can I do this? I don't know. Right above the methionine are the letters ATG. That is actually the cDNA, the cDNA codon for methionine, right? So all of this, see how all of that's just a whole lot of Gattaca nonsense. It's not nonsense. The arrangement of those makes it so that there's another machine we're going to talk about can read all of that G, 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 A, G, C, G, 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 C, T, all of that, and it knows, oh, we need an arginine and a glutamine and an arginine and a glycine. It knows because that is the code, right? Oh, you need to know this. This is probably going to literally be on the quiz. The sequence of codons in messenger RNA. This is not messenger RNA. That's something that scientists use. You don't have to know that. But the sequence of codon in the messenger RNA or the sequence of codons in the DNA ultimately determines the order of amino acids in the proteins, the sequence of the codons. And each codon is a little code word that tells the machine that reads it which amino acid to put next. That two-part molds, yeah, we got that. Okay. Now, DNA has got two really big jobs. And I'm going to start there at the beginning of the next video.